Great. Thank you, Firaj, for the kind introduction. And I go by Barathi, and I'm a scientist in drug product development, biotherapeutics drug product development at Janssen, and based on Springhouse, Pennsylvania. And today I will present some challenges and opportunities in cell therapy formulation and delivery. So in our organization, we are responsible for the drug product development, and we work with many biologic modalities. We work with traditional MABs, bispecifics, and conjugated antibodies, and a variety of gene and cell therapy products. Particularly, we are very proud of our cell therapy portfolio in discovery and development. We filed BLA and MAA and numerous other health authority filings 6 months ago for our first cell therapy product. It is against BCMA. It's an anti-BCMA autologous CAR-T product. And we are working to answer the questions and hoping to get the approval soon for commercialization. And also we have, and that's Cilta-cel, the anti-BCMA product. And we have also a collaboration with Fate Therapeutics to develop up to 8 allogeneic cell therapy products. These are iPSC-derived products, and we are trying to file an IND soon in USA. So today, my talk is pretty much divided into 3 sections. I will give a brief introduction to the cell therapy drug product development. And I'll present 2 case studies today, one around formulation development for cryopreserving certain allogeneic cells. And also the other one, the second case study is screening excipients for improving the in-use properties of some autologous cells. So that's going to be my focus today. I will start with a brief introduction to CAR-T cells. So CAR-T cells, the chimeric antigen receptor, the CAR, it contains an extracellular scFp, which is derived from the antibody, which confers affinity and specificity for the CAR. And it has intracellular cytotoxic and co-stimulatory domains, which are derived from the T cell receptor, and which is known for its cytotoxicity and tumor killing abilities. So, and this chimeric antigen receptor, it is transduced into T cells, and the T cells become the CAR T cells. And very briefly, the mechanism of action. So the scFv of the CAR-T recognizes the tumor target, and so the recognition initiates the downstream signaling pathways, and which leads to the release of perforins and gramzymes from the CAR-T cell, and then the tumor cell in the vicinity it's get killed and lysed by the molecules. So that's a very basic mechanism of action. So the CAR-T or cell therapy products in general are broadly classified into 2 types in terms of manufacturing: autologous CAR-T and allogeneic CAR-T. So for autologous, the T cells are actually obtained from the patient. So the starting material is obtained from the patient. The CAR construct is transduced into the patient-derived T cells, typically by a lentivirus. And then it's expanded, and it's formulated, fill-finished, frozen down, and then sent to the patients for infusion. And it's a patient-specific product. So one patient, one product, and what we call as vein-to-vein time from or the needle-to-needle time, it typically takes up to 4 weeks. And in terms of allogeneic products, we have the cells, the starting material, typically from a healthy donor, or it's stem cell derived, it's iPSC derived, and those cells are genetically engineered using a CRISPR-Cas9 or any stable genetic engineering methods to knock out the unwanted genes and knock in the desired genes and make a cell bank. And those cells are pretty much expanded, purified, full finished, and then it can be infused to any patient. So it's an off-the-shelf product. So that's allogeneic cell therapy products. As we know, we have 5 products approved in the States, 5 CAR T, autologous CAR T products approved in the States for commercialization. And but there are a huge number of molecules in clinical development. We have T cells, NK T cells, or TCR products, or macrophages, and we have numerous cell types across different phases of development. And so also we're going to have numerous advancements in the cell therapy technology over time. What I have on the right here is a innovation S curve from the publication mentioned below, and so which describes the path for cell therapy development innovation for the next 10 years. And so we are at first generation right now, and we are going to see enhanced safety products with enhanced safety, off-the-shelf products, and combination therapies, and products for solid tumor and precision oncology. The next 10 years. Now, I like to start, getting into the drug product development. I would like to start with comparing our well-known antibodies, the proton pharmaceuticals, versus our cell-based medicinal products, right? So, we've been working with MAPS for 30 years, and so we are very comfortable with them, and we have evolved over time. We're like small molecules, like only 10 nanometers relative to the cell-based pharmaceuticals, which are 10 micrometer or above. Like T cells are 30 microns, and we know, we filed many applications, and we've manufactured many products, and we have so we know the product and process in detail, and we know the end-to-end automation, and we have better understanding of the whole MAP process, right? So and again, it's a discontinuous process. We have a frozen drug substance, and it's the formulation components are well known. It's typically in a vial or pre-filled syringes. It has a long shelf life, even at refrigerated temperatures. The use time is longer. It supports multiple routes of administration. Whereas in terms of cell therapy products, like you mentioned, it's a living drug product. So it's much larger. It's a living drug product, and typically it sells in suspension. So it's a infusion bag containing cells in suspension, and the formulation is very complex for cryopreservation. So it's typically stored at minus 120 or lower. It's typically a vapor phase liquid nitrogen, and the excipients, mechanisms of action are not well characterized, and it has a very short shelf life once thawed, because the formulation contains a DMSO that's toxic to the cells, so it's only a couple of hours longer, and it's typically a IV route of administration. It's an intravenous route of administration, right? And again, this is a relatively new field, so we still have good guidelines on the impurities, particularly when it comes to the clumps or the particles in drug product, and also this is a living drug product, so it's very sensitive to any small changes or interactions, and the manufacturing process is continuous. So we unfortunately don't have the luxury of having a frozen drug substance. The drug product process typically takes place within a couple of hours, and it's done on the same day of harvest. And there are a lot of manual steps and open steps, right? So, and the automation is its infancy, and most importantly, we cannot filter the product at the end of the process, right? Because it's it's larger than 0.2 microns. So that's very important to make sure that we have a sterile process, and we cannot filter at the end of the process or even before infusion using a 0.2 micron filter. So and so that's pretty much summarizes the differences between our known MAPs and our cell-based pharmaceuticals. So this slide summarizes the dosage and administration of 4 out of the 5 marketed CAR-T products, right? And so these are typically a few million cells in an infusion bag. Except for Breyanzi. Breyanzi is the only product that actually comes in vials, whereas all the 4 other products come in infusion bags, and it's typically from 10 to 70 ml. And again, I wanted to highlight that the products or the shelf life is the post-thaw in use time, or the shelf life is actually much shorter, some 30 minutes to 3 hours. And they have different methods of thawing or infusion methods, the flush procedures, the filter requirements, and procedures to deal with cell clumps upon thawing. So again, these are some product development variables that need to be well understood in advance to push the product further in development. The one example, what we found out is that the presence of clumps may pretty much will determine the method of administration, because if we are choosing to go with a syringe push, like an IV infusion, a syringe push, the product must be free of clumps or visible particles, because there are no FDA-approved non-leukocyte depleting filters to filter the product and administer using a syringe. So typically that is for those products with clumps, we need to have a filter, a non-leukocyte depleting filter, which is supported by infusion bags and IV administration sets. So the point is, so this field is still in infancy, and we don't have a lot of guidance, and so we have to make sure that we have a better product with a high quality to better suit different indications and different routes of administration. Moving on, I would like to highlight the different drug product processes, the steps in cell therapy. So it typically starts with washing the API, the drug substance, harvesting and washing the cells on the same day as it's harvested. Typically, it's a 10 to 40 day process based on the cells, the starting material. So the harvest day is pretty much the whole drug product process happens on the harvest day. So it's harvested, washed, and formulated with a formulation buffer that's typically for cryopreserving the cells at the end, and it's fill finished in bags or vials, and goes a visual inspection. Typically, it's manual, and the automation platforms are still in infancy, particularly for bags. Right, we have some. There are few vendors for vials. Again, we have to develop these methods. I mean, we are still, particularly for scale-up processes, like for large-scale processes, it's very hard to find different equipments or different vendors to suit all the unit operations in the drug product process. And again, labeling of vials may not be possible after cryopreservation. And cryopreservation typically happens using a controlled rate freezer, and following which the vials or bags are stored in vapor phase liquid nitrogen and then shipped to the clinical sites or the pharmacy. So again, it's actually the whole process must be completed in a few hours, because as soon as the cells are exposed to DMSO, the clock starts ticking. We only have an hour or so to complete to put them in liquid hydrogen storage. Right. Again, in Anson, we are at a point where we have both autologous allogeneic products. So and we are scaling out the autologous process, and we are scaling up the allogeneic process, right? And what we find is, as I mentioned, particularly for scale up, there are not a lot of choices in terms of different equipment from different vendors for all these unit operations, particularly backfillers or visual inspection stations or high throughput cryopreservation solutions. And again, we are working to build a end-to-end large scale high throughput platform for our FADE collaborative allogeneic products. And we are trying to close the process for our autologous platform. We are trying to close the process and remove a manual touch points and exert a full control over the autologous process and have a robust, a consistent process. So it's all over the years we have a good process for MAPS, whereas in cell therapy we are, the field is trying to build a robust process, and because we are starting with, if you remember, we are starting with the raw materials are not the same, right? Particularly for autologous products, we the patient's T cells are variable. Like one product is from the other product, so we pretty much release product one by one, and so the product must be under good control and robust enough to suit different patients and also across different populations and different indications. So again, the field and also Janssen is working to build a robust autologous allogeneic cell therapy processes. So moving on, I'd like to highlight some challenges and opportunities in cell therapy product development. I think I touched on a few points. They are in 4 different buckets here. In formulation, like I mentioned, the field is trying to stay away from DMSO. So the field is looking at developing low DMSO or no DMSO solutions and understanding the mechanism of action of the infusions, infusion limits, having different options for container closure, and understanding having a deeper understanding of the product under normal and stress conditions, different analytics, and how can we store at higher temperatures, in a mechanical freezers or such. So there are a lot of questions in this field, in this evolving field. Again, the same goes for the lentivirus, right? It's intermediate drug substance, and particularly for autologous products, the delivery platform also needs to be formulated, fill finished, and should be controlled for making for the final drug product. And I think I highlighted a few challenges in process development. And finally, the delivery aspect of it, right? So the storage, shipping, handling, and thawing of the cell therapy product. Administration, we don't have standard operating procedures. So we have different procedures across sites, across countries for administering cell therapy products, different filters. The blood sets come with different materials of construction, and they have a shorter shelf life and in use time. And also the field is looking at alternative routes of administration. So and those alternative administration routes typically cannot support DMSO. Like a lot of products are for intracerebral delivery. A lot of clinical trials have cell therapy products for brain tumors, and it's a very highly concentrated 2 ml product without DMSO. So and again, we are also looking at combination products. Right? So the field is it's like fixed dose combinations, putting cells and antibodies or cells and small molecules in the same vial, and then there are at least 3 such products, and one of them are good. One of them actually has very good phase 1 results. So I think the drug product community should also be evolving at the same speed to support all these novel cell antibody or cell small molecule combination products. And again, these are not sequential infusions. This is like a product, a vial that contains both the products in the same vial. So, and so there are a lot of technologies and innovation also in terms of biomaterial mediated delivery. Again, supply chain and COGS is always an interesting aspect of cell therapy. Right. So again, finally, I would like to highlight the importance of an integrated drug product design in cell therapy. So it starts with understanding a deeper understanding of the molecular properties, the biology and the biochemistry of the molecules, and finding the right formulation, buffer, and the processes, and studying the impact of the process on the product, and fitting the process to the plant, and understanding the manufacturing distribution networks, because we get the T cells from the patient, and also it's the patients are waiting for the product, right? So we must know the distribution networks and the supply, the clinical supply needs, and also the commercial supply needs well in advance to make the drug product. And also, again, we make the products for the patient. So we should understand the patients, particularly for this therapy, since the starting material is actually from patients. We need to know the health, the T cell health, and the indication, the stage of the patient, and also how the patients and healthcare providers are interacting with each other, and all these interactions and the phase of the development, they're all important to deliver the better drug product and to meet patients' needs, right? So with that, I will move on to my case studies. And given we only have 10 minutes, I will try to just highlight the important points in both my case studies. So very, very briefly, the principle of cryopreservation in cell therapy is a controlled and reversible dehydration. So the water is removed from cells in an optimal manner. So I'm using a combination of excipients and a controlled rate freeze. And the excipients can be intracellular, such as DMSO, or extracellular, such as sugars and polymers. So and then the cells are stored in a vapor phase liquid nitrogen in a metastable state for up to 9 to 12 months. And then upon thawing, the cells must retain the water back for proper function. And again, this is a very a short study to find the right formulation conditions for cryopreserving some allogenic cell types at 2 different temperatures for up to 90 days. And we used untransduced primary cells into healthy donors. And then we tested 6 excipients with 6 different mechanisms of action. And then we had we had total 13 conditions. And again, I won't go into the details given the time constraints. So first, we set a benchmark. So we used a commercial cryopreservation buffer, and that contains a DMSO. And we show that typically the cells have 80%. The health of the cells are 80% up to 3 months, and beyond which it starts to deteriorate over time. And again, this is a flow cytometry-based detection. And this slide summarizes our formulation screening results from the studies on both cell types, 2 different conditions. The y-axis has % healthy cells, and we have 13 different conditions in the commercial condition as a control. So as you see, there are a few conditions, 3, 4, 5, 7, 9, and 11 work similar to the commercial formulation. And some of them contain DMSO, some don't. So we have done doing some statistics to find the right excipients and combinations to test them further. And again, we can use advanced analytics. We can look at specific subsets using flow cytometry and study the impact of formulation on these specific cell sets, subsets of cells. Again, this is one example to show that we are also screening some alternatives to DMSO, because for DMSO is intracellular cryoprotectant. To replace DMSO, we need to screen for excipients that can actually penetrate cells. So we are actually setting up high throughput imaging assays to find the right excipients that can be an alternative to DMSO. So in summary, this work was designed to show that we can set up some small screens, targeted screens, to find the right formulation buffers and conditions for cryopreserving allogeneic cells, possibly without DMSO. And we are trying to optimize these conditions further using functional and biological assays. So my next case study focuses on developing or increasing the in-use time of these cells. Right. So obviously, it's better to have a formulation without DMSO to improve the shelf life of cells. And also, but in the short time, we wanted to see whether we can improve the DMSO containing formulation, because in most cases, what we found out is that we need more than 2 to 3 hours of post-thaw in-use time for these cell types. So we set up a small study. It's like a controlled infusion, a simulated infusion study to see whether we can add a certain excipients with known mechanisms of action to this commercial DMSO containing formulation buffer to increase the in-use time of the products post-thaw during infusion and at the clinical sites. So again, I won't go into the details of the study, but again, this is a standard simulated infusion experiment. What you could see here on the left is that the left graph, the control is the commercial formulation buffer, and the y-axis is % recovery post-thaw. So this is a stress condition. So we held the drug product in the administration set for up to 3 hours, followed by a 1 hour simulated infusion. You could see certain excipients, addition of certain excipients to the commercial buffer can increase the recovery up to 100%, particularly for a low dose drug product, right? And again, we show that this is also possible. We can add these excipients pre-freeze during the formulation, and we can still see some excipients increasing the recovery of the low-dose drug product. Right? We also show that not only the % recovery, is increased with certain excipients, also the quality of the product remains the same with some excipients, particularly for excipient 1. We don't see any dead cells, right? Or we don't see any necrotic cells or late apoptotic cells. All the cells are pretty much retained as early apoptosis phase. So upon infusion, they can revert back to healthy cells. So these excipients not only increase the recovery, also try to maintain the product quality over time, right? So to summarize, this second case study shows the importance of the in-use time in cell therapy, and particularly in some cases we see that the drug product is thawed in one site in one building, and then it's actually not thawed on the bedside. It's actually thawed in a cell lab or a pharmacy and moved to a different building. It takes 40 to 45 minutes for moving the thawed drug product from one place to other. And if it's more than 45 minutes, and then the shelf life is only 30 minutes with DMSO, I mean, we must increase the shelf life of the product. So we show that holding the drug product in the infusion set in some cases can be detrimental, and that can be resolved with the help of certain excipients and adding certain excipients to the drug product. So, I mean, in closing, I'd like to summarize my talk. So I think I started with a brief introduction to the product development, drug product development in cell therapy, and the importance of integrated drug product design in cell therapy. And then I presented 2 case studies, and one developing formulations for cryopreserving allogeneic cells, and one to selecting right excipients for increasing the shelf life of the drug product. So potential solutions, some challenges and opportunities in cell therapy product development. And I'd like to thank the team and the leadership and take any questions. Thank you.